Synthetic biology, or SynBio, is an interdisciplinary scientific field that combines engineering and biology to create new or redesigned biological components, devices, or systems which do not naturally exist. It draws from a myriad of scientific domains such as biotechnology, genetic engineering, systems biology, and even electrical or computer engineering. This scientific endeavor aims at designing and constructing biological pieces, systems, and machines, or revamping them for beneficial purposes. Putting an emphasis on infusing engineering capabilities into organisms to adapt their functionalities according to need is another key focus of this field. It applies engineering design principles to biological systems to produce robust and predictable systems with novel abilities as per the European Commission. This might involve the use of molecular assemblers like ribosomes. The origination of this branch of science could be traced back to 1910 with the first known use of the term synthetic biologies by Stephane Ledouce in his publication. This was followed by significant milestones in the field. In 1944, Oswald Avery's research confirming DNA as the genetic material laid the foundation for future genetic research. The iconic publication on DNA structure by Francis Crick and James Watson in 1953, and the postulation of cellular regulation by molecular networks by Jacob and Monod in 1961, all added to the growing scientific knowledge. The dawn of synthetic biology came in 1973, with the first molecular cloning and DNA amplification in a plasmid executed by Cohen, Boyer et al. This paved the way for the new era of synthetic biology, where not just existing genes were analyzed, but new gene arrangements could be constructed and evaluated. This period also saw the invention of biobrick plasmids, the widely used standardized DNA parts. In 2003 today, synthetic biology continues to evolve, broadening the horizons of biological engineering and design. In the realm of synthetic biology, scientific progression over the years has yielded remarkable results and continues to chart new territories. 2005 witnessed the emergence of a light-sensing circuit in E. coli, and the formation of multicellular pattern circuits progress continued in 2006, with a synthetic circuit aiding bacterial invasion of tumor cells marking a milestone. A significant breakthrough in 2010 saw the first synthetic bacterial genome, M. mycoides G. CV Synone.0, published in Science. The genome, synthesized from chemicals using yeast recombination, was the first of its kind. Following this, 2011 allowed the engineering of functional synthetic chromosome arms in yeast. In 2012, CRISPR-Cas9 bacterial immunity underwent programming for DNA cleavage, a tremendous advancement in gene editing. In 2019, a milestone in synthetic biology materialized with the first computer-created bacterial genome, Caulibacter ethensis 2.0, by ETH Zurich scientists later that year. The production of a synthetic, viable form of the Escherichia coli bacteria was profoundly revelatory with regards to understanding life-form modification. The following year, 2020, heralded the birth of the first xenobot, programmed from frog cells using AI, further solidifying synthetic biology's potential. By 2021, these xenobots demonstrated self-replication capabilities, thus adding another tier to the evolution of synthetic biology. In essence, synthetic biology is an exponentially growing field aiming to enhance and redesign biological systems for a variety of purposes like information processing, chemical manipulations, energy production, and human health improvement. Engaging about 350 companies across 40 countries, the global net worth of synthetic biology-related work is estimated to be $39 billion. With no universally recognized definition yet, it is broadly perceived as the fusion of genetics and physical engineering to create new life forms and characteristics using chemically synthesized DNA. The field advances biotechnology, offering solutions to challenges in various sectors like agriculture, manufacturing, and medicine. Through genetic engineering, the creation of synthetic chromosomes or minimal organisms is possible, aiming to introduce new technology functions in living cells. In the realm of synthetic biology, there are 5 distinct areas: bioengineering, synthetic genomics, protocell synthetic biology, unconventional molecular biology, and in silico methods. Understanding the nuances and overlaps between these categories holds a crucial importance in the context of social and ethical evaluations, as well as for calling attention to global versus unique issues. Primarily driven by the ambition of asserting biotechnology as a recognized engineering discipline, bioengineering stands as a pioneering subfield. It's noteworthy that the term bioengineering here is distinct from conventional genetic engineering, which involves transmitting a single transgene to a target organism. Bioengineering within the scope of synthetic biology presents a wider, more intricate perspective on modulating organisms or metabolic structures. A classic example of single-gene embedded genetic engineering is how bacteria are integrated with human insulin gene to produce transgenic protein. However, under the umbrella of synthetic biology, bioengineering embarks more elaborate ventures, like forming new signaling pathways encompassing multiple genes and supervisory elements. A case in point is conceiving an oscillator circuit to stimulate systematic GFP, green fluorescent protein, output in mammalian cells. By applying metabolic and executive modules and other standardized components, the ambition of bioengineering is to design unique biological systems. The approach is expected to render bioengineering more manageable and predictable than its traditional counterpart, unlocking limitless possibilities for innovation. Synthetic genomics brings to light another facet of synthetic biology, majorly manifested in the creation of beings with chemically engineered minimal genomes. Such advancement is due to the continuous progression in DNA synthesis technology, which now allows reasonable production of DNA molecules with thousands of base pairs. The aspiration is to interlace these molecules to form wholesome genomes and transplant them into live cells in place of the host cell's genome, thereby reprogramming its metabolism to exhibit different functions. This method already demonstrated its potential when scientists successfully created infectious viruses by synthesizing multiple virus genomes, raising the initial concern about potential risks. The simplistic genome has the potential to be a chassis genome that can expediently expand through the inclusion of genes manufactured for specific purposes. The pursuit of synthetic biology, specifically the creation of protocells, aims to synthetically create the intricate components that make up a living cell. These manufactured cells made from lipid vesicles packed with essential building blocks like enzymes and nucleic acid are intended to perform like an actual cell, being capable of self-replication, self-preservation, and evolution, advancing our understanding of the underlying requirements for life to exist. Protocell synthetic biology is an exploratory venture, however. It could offer practical applications, such as creating bioproducts or drugs. A more experimental approach within synthetic biology is unnatural molecular biology, which seeks to construct new types of life defined by modified nucleic acids or a novel genetic code. These extraordinary life forms are made possible by altering the conventional DNA and RNA structures or by manipulating the normal genetic code to produce new and unique proteins and amino acids to achieve this. A significant challenge lies in adapting a cell's enzymatic machinery. This revolutionary form of life comes with both potential advantages and risks. One notable benefit is the prevention of genetic mixing with natural species, as these synthetic organisms are designed to require non-natural nutrients, reducing the risk of them surviving in the wild if accidentally released. However, if these engineered life forms adapt and thrive in uncontrolled spaces, they could potentially outcompete natural organisms as they'd be resistant to conventional predation and viruses. This could potentially lead to an uncontrolled proliferation of these synthetic organisms. Synthetic biology, as a unified field, systematically engages with 5 core areas, with each exploring different aspects of life. The areas of study include metabolic regulation, essential elements, and biochemical composition, all aiming towards the common goal of creating new life forms. The methodologies adopted in these areas are diverse, contributing to the wide-ranging approaches within synthetic biology. The discipline of synthetic biology is inherently interdisciplinary, synthesizing knowledge from a multitude of sciences. The central ambition of synthetic biologists, regardless of the multitude of methodologies and tools they use, is the design and production of new life form. Therefore, any evaluation of synthetic biology, be it ethical, legal, or safety considerations, should account for its overarching aim while also addressing unique questions and risks inherent to various techniques. The 4 conventional engineering approaches applied in synthetic biology are top-down, parallel, orthogonal, and bottom-up. These approaches employ unnatural chemicals to recreate biological behavior and construct artificial life. They also use interchangeable components from natural biological systems to construct non-natural systems. Such synthetic objectives necessitate researchers to explore new territories to tackle complex issues, driving new paradigms beyond analysis reach. In the process, synthetic biology has already resulted in inventions like diagnostic tools that greatly improve treatments for infectious diseases and devices that oscillate, creep, and even play tic-tac-toe. This field also implements metabolic and genetic engineering techniques to introduce new functions to living cells. The aim is to reduce cellular complexity by comparing universal genes, removing non-essential ones, and developing simpler genomes, all under the assumption that there existed a last universal common ancestor that led to all life forms. Recent research, however, suggests that life might have originated from a group of cells rather than a single one, complicating the quest for the minimum genome. Trimming non-essential functions tends to weaken an organism and results in delicate genomes. Finally, synthetic biology endeavors to produce new biological systems in vitro by combining non-living biomolecular components, often with the goal of creating an artificial cell. The amplification of synthetic pathways in an artificial setting contains significant potential impact on other sectors within synthetic biology, like metabolic engineering. It's important to re-emphasize this research as essentially synthetic biology, since it is no longer classified as such. This focusing on bioengineering or parallel engineering lies at the very heart of biological systems. The research uses foundational elements like the basics genetic code, conventional biomolecules such as nucleic acids, and the 20 standard amino acids to design these systems. The engineered elements then have broad applications in diverse fields, such as biocomputing, bioenergy, remediation, optogenetics, and medicine. Most of these applications rely on the usage of vectors or plasmids, small circular DNA units that can replicate separately from chromosomal DNA. From another perspective, a different approach is taken, referred to as chemical synthetic biology or perpendicular engineering. It aims to modify or enlarge the genetic codes using artificial DNA bases and artificial DNA bases nor amino acids. It ties in closely with xenobiology, a relatively new field that fuses synthetic biology, systems chemistry, exobiology, and origins of life research. This approach has led to researchers inventing compounds similar to DNA bases to see if these alien molecules could carry genetic information. They've also replaced the DNA sugar, deoxyribose, with non-canonical units. The theme of this approach centers on the innovative concept of expressing information beyond that of the standard 20 amino acid proteins by altering or expanding the genetic code. One method incorporates a specific unnatural or xenoamino acid, XAA, into specified proteins at pinpoint locations by using orthogonal enzymes and an RNA adapter from another organism. Through a process of directed evolution, varying the genetic sequence can lead to the derivation of orthogonal ribosomes. This eases the integration of XAAs into proteins or enables the creation of mirror life, systems containing biomolecules with differing chiral orientation. Precision in computations in design is achieved through analysis under diverse conditions. The field of synthetic biology has seen remarkable advances due to cost reductions in oligonucleotide synthesis and the discovery of pre-sure DNA structures derived from oligos have expanded to a genomic scale, with the synthesis of larger genomes like hepatitis C virus in 2000, poliovirus in 2002, the bacteriophage Phi-X174 in 2003, and the synthetic genome of Mycoplasma laboratorium in 2006. By 2007, companies were even offering genetic sequences, synthesis of up to 2,000 base pairs for economical prices and rapid turnover. Advancements include the use of photolithographic or inkjet manufactured DNA chips, KIR and DNA mismatch error, correction for cost-effective large-scale genetic modulation to enhance gene expression or infuse novel amino acid. The emergence of CRISPR-Cas as a practical method for gene alteration has been called the biggest breakthrough in almost 3 decades in synthetic biology. Thanks to CRISPR, what once took months or years now only take weeks. In synthetic biology, DNA sequencing plays the triple role of boosting knowledge about natural organisms, verifying fabricated systems, and speeding up the identification of synthetic organisms and systems. BioBrick plasmids, invented in 2003, are the most frequently used standardized DNA elements. Stored at the Registry of Standard Biological Parts, BioBrick plasmids have facilitated global student participation in iGEM competition. However, overlapping genes pose a challenge to manipulation due to sequence overlap between genetic elements, emphasizing the necessity for their independence. The study of protein-protein interactions within cells is crucial, particularly when it comes to chemically induced dimerization or the utilization of light-sensing domains. In living cells, molecular motifs exist within larger networks, which can impact the functionality of the nucleus. This effect may vary in isolation versus within the integrated network. Improved predictive models contribute significantly to synthetic biology, enhancing our understanding of key cellular activities and providing a more comprehensive insight into cell behaviors, gene regulatory networks, and molecular interactions in general. Extensive modeling is instrumental for exploring the dynamics of gene expression, an intricate web of complex relationships which are central to biological research and design. Dynamic simulations allow for detailed blueprinting of molecular interactions involved in key cellular activities. Microfluidics, more specifically droplet microfluidics, has emerged as a vital tool for the construction and characterization of new components, extensively used in various screening assays. Attention has also been given to understanding the DNA transcription mechanism in the quest for controlling synthetic DNA transcription in both unicellular and multicellular organisms. Studies have demonstrated the adaptability of synthetic transcription factors. The transformative power of synthetic biology has led to its utilization in various applications, such as bioremediation through microorganisms, the production of beta-carotene using modified rice, and even the generation of rose oil using genetically engineered yeast. This diverse range of applications not only promotes sustainable practices, but can also potentially address health issues such as vitamin A deficiency, affecting hundreds of thousands of children annually. What is multifunctional usage of biosensors in the detection of pathogenic signatures such as SARS-CoV-2, as well as their potential use as wearable devices? It delves into the complex regulatory circuits that cells have developed in response to various environmental factors, including transcriptional to post-translational circuits. These circuits comprise transducer modules that filter signals and trigger biological responses, as well as specially designed sensitive sections that regulate the attachment of analytes and control signal detection thresholds. The modularity and selectivity of these biosensor circuits are coordinated at the transcriptional, translational, and post-translational levels, delicately balancing the two key sensing modules. The paragraph also notes the synthesis of products other than animal foods, like the commercialization of synthetic coffee. This same technology is also being utilized to produce photosynthetic microbial cells for the creation of synthetic spider silk, highlighting another potential application of synthetic biology. Furthermore, the text discusses the breakthroughs made in biological computing, an increasingly popular field in synthetic biology. Researchers have successfully developed various logic gates in many organisms, demonstrating analogy and digital computations in living cells. Advances include proof of concept therapy that uses biological digital computation to target and eliminate human cancer cells, automation of digital circuit design in bacterial cells, and the engineering of digital computation in human cells through a system named Boolean logic and arithmetic through DNA excisions, BLADE. Lastly, the paragraph discusses how cells use interacting genes and proteins, referred to as gene circuits, for various functions such as responding to environmental signals, decision-making, and communication. Synthetic biologists have engineered these gene circuits to control gene expression at multiple levels, including transcriptional, post-transcriptional, and translational levels. The process of re-engineering has led to the production of curly fibers, an essential component of biofilm's external layers, which can serve as a base for programmable nanomaterial. By genetically tailoring these nanofibers, they can perform specific functions like adhering to substrates, templating nanoparticles, and immobilizing proteins. For instance, with the help of directed evolution, a method of protein engineering, novel protein structures that exemplify or augment the functionality of existent proteins can be created. A group managed to create a helix bundle capable of binding oxygen similarly to hemoglobin, without the ability to bind with carbon monoxide. A separate group produced G-protein receptors that could be activated by the inert molecule clozapine N-oxide, but were immune to the effects of the native ligand, oxetylcholine. These receptors are known as DREADs. In addition to this, Functionalities and protein particularity can be engineered using computational techniques. In a specific study, a bioinformatics and molecular modeling process was used to sequence databases, and a computational enzyme design method programmed enzyme specificity. The result was designed enzymes with over 100-fold specificities for producing longer-chain alcohols from sugar. Scientists also typically explore the expansion of the natural 20 amino acid set. Despite the identification of 61 codons, excluding stop codons, only 20 amino acids are generally coded across all organisms. Therefore, certain codons are engineered to code for alternative amino acids, including non-standard varieties or exogenous types. The engineering of protein structure and function is also gaining traction through the reduction of the normal set of 20 amino acids. One such investment saw the use of an engineered version of chorismate mutase, which retained catalytic activity even when composed of only 9 amino acids. Synthetic biology is employed by researchers and companies alike to create industrial enzymes that deliver high activity, optimal yields, and improved efficiency. For example, this has been applied in the improvement of everyday products like detergents and lactose-free dairy products, and in making them cost-effective. Synthetic biology represents a prime example of the application of cutting-edge biotechnological techniques in the industrial sector, having wide-reaching implications in areas like pharmaceutical discovery and the optimization of chemical fermentation by investigating modular pathway systems in biochemical production. Synthetic biology can boost the yields of metabolic production. Therefore, the artificial activity of enzymes and its impact on metabolic reaction rates could lead to novel strategies to augment cellular properties in various industrial applications. Technological advancements in the field of synthetic biology have led to revolutionary developments, particularly in the incorporation of unnatural nucleotides and amino acids into nucleic acids and proteins. One remarkable achievement took place in May 2014 when scientists successfully infused 2 artificial nucleotides into bacterial DNA. Although their efforts to generate mRNA or proteins utilizing these artificial nucleotides were unfruitful, they made significant progress by successfully exchanging the bacteria 24 times. Synthetic biology has garnered attention from NASA due to its prospective contribution towards resource production for astronauts using a limited range of compounds sent from Earth. The prospect of utilizing this discipline to create production processes based on local resources on Mars has been especially intriguing. Consequently, efforts are being made to develop plant strains that can survive harsh Martian conditions, employing techniques similar to those used to bolster agricultural crops' resilience against various environmental factors. A key area of focus within synthetic biology is synthetic life. Scientists are eager to create hypothetical organisms from biomolecules or their chemical analogs, either in the lab. The goal is to recreate life from non-living, abiotic components or to better understand life's properties and origins. Furthermore, synthetic life biology endeavors to create living organisms capable of performing essential functions ranging from pharmaceutical production to detoxifying pollution, stricken land and water. Despite these advancements, a completely synthetic cell possessing the ability to capture energy, maintain ion gradients, store information, accommodate macromolecules, and exhibit mutative tendencies remain elusive. The closest success story was recorded in 2010 by Craig Venter's team, who developed a completely synthetic bacterial chromosome that they implanted into genetically cleared bacterial host cells. Notably, the hosts managed to grow and replicate, making Mycoplasma laboratorium the first and only organism with a completely engineered genome. In 2014, a team heavily altered E. coli's genome, replacing it with an expanded genetic code chromosome. This method enabled the creation of the first living organism with artificially expanded DNA code. Another noteworthy development took place in 2019 when researchers managed to create a viable variant of the bacteria Escherichia coli with a reduced codon count in its genome. The year 2017 marked the beginning of the large-scale international build, a cell research collaboration aimed at creating synthetic living cells, followed by national synthetic cell organizations in various countries, such as FabricCell, MaxSymbio, and Bicycit. The European Synthetic Cell Initiatives unified under the SynSalu banner in 2019. The therapeutic treatment of cancer often sees the use of conventional medications that indiscriminately target both tumors and normal tissues. Hence, artificially engineered viruses and organisms, which can specifically identify the pathological signals and fine-tune their therapeutic action, may prove to be more advantageous. For instance, 53 pathway activity was installed into adenoviruses to regulate the replication process in human cells. The application of bacteria in cancer treatment has been a long-standing practice, using bacteria such as Bifidobacterium and Clostridium that selectively colonize tumors to decrease their size. Synthetic biologists have made significant strides in reprogramming bacteria to detect specific cancer states and respond accordingly, further mitigating off-target impacts. Various approaches have been undertaken to direct the therapeutic molecules straight to the tumors. For instance, making bacteria respond to the tumor's microenvironment, such as hypoxia, is one method. The other approach incorporates peptides that can specifically identify a tumor when expressed on the surface of bacteria. Furthermore, numerous types of bacteria have been utilized in cancer therapeutics, including Salmonella typhimurium, Escherichia coli, Bifidobacteria, Streptocoli, Bifidobacteria, Streptococcus, Lactobacillus, Listeria, and Bacillus subtilis. All these bacterial species have unique properties and make distinctive contributions to cancer therapy. Similarly, synthetic biologists are creating genetically modified live yeast to serve as microfactories in producing therapeutic biologic medicines. Live yeast has the principal advantage of being eukaryotic, thus can be administered together with antibiotics. Turning our attention to the immune system, it has a vital role to play in cancer and can be leveraged to target cancer cells. This has resulted in a focus on cell-based therapies, primarily involving the engineering of T-cells. Zeta Inc. has proposed the first in-human clinical trial of engineered live yeast for treating Clostridium difficile infection by 2024. Corn-based ethanol, the most common biofuel, faces difficulties due to its high production cost and less than optimal fuel characteristics. The answer may lie in genetic engineering of microbes to efficiently transform biomass to biofuels. However, this solution depends on their production costs competing with or exceeding those of existing fuel production. Similar cost-related issues affect the manufacture of several medicines, limiting their broader therapeutic use. The introduction of new synthetic biology tools could significantly benefit both the fabrication of new materials and microbiological manufacture of biomaterials. A notable method is the CRISPR-Cas system that's noted for its simple, modular, and scalable approach to genome engineering in diverse organisms. Pivotal components to consider in constructing and refining biological systems entail regulators, ribosome, binding sites, RBSM, terminators. Further development in synthetic biology is also seen in lab-grown organoids, demonstrating potential for medical research and transplant applications. One of the most promising technologies is 3D bioprinting, used in various medical fields including organ reconstruction. Noteworthy cases, such as an infant with a rare respiratory condition treated using a 3D-printed tracheal splint and patients with end-stage bladder disease receiving engineered bladder tissue replacements, underscore the scope of this innovative technology. However, significant challenges remain, including the complexity of printing an entire organ with intricate shapes and structures. As an example, a bioprinted heart must fulfill not only structural requirements, but also vascularization, mechanical stress, and electrical signal propagation specifications. So far, limited success has been achieved in printing fully functional organs. Investigation and advancements in synthetic biology are focusing on the development of regeneration methods for humans, along with the fabrication of transplantable artificial organs. Synthetic biology is also lending to the creation of nanoparticles, useful for drug delivery and other purposes. This research is now being furthered with the creation of synthetic cells which replicate the functionality of biological cells, with potential uses in various fields such as medicine. For instance, designer nanoparticles can eradicate portions of atherosclerotic plaque, a leading cause of heart attacks, from the interior of blood cells. Additionally, synthetic microdroplets for algal cells may produce hydrogen as a byproduct, offering potential biotechnological benefits in a hydrogen economy. However, as synthetic biology intrudes into the fabric of life, it brings along an array of ethical questions. Concerns like misuse, control, and access, and the benefit distribution of synthetic biology become prominent. Pondering the patent system's stance on living organisms and determining the moral or legal status of new creations are considered important ethical aspects. These issues are further elaborated by three main features: biosafety, biosecurity, and the creation of novel life forms. Other considerations include the regulatory mechanisms, patent management of new creations, the equitable disbursement of advantages, and research integrity. The ethical dimensions of synthetic biology are not unprecedented, as similar issues have been raised in the past for recombinant DNA and genetically modified organisms. Former head of the Presidential Bioethics Commission, Amy Gutmann, propagated caution against excessive regulation of synthetic biology and genetic engineering. Gutmann emphasized that over-regulation can hinder innovation, negatively impacting safety and security by obstructing the development of efficient safeguards. One of the key ethical dilemmas involves the acceptability of creating new life forms, often dubbed playing God's. While the creation of artificial life forms is currently on a small scale, their potential benefits and risks are yet to be fully understood. Advocates argue that creating artificial life forms could contribute significantly to various fields, including agriculture, medicine, and academia. However, concerns linger that such interventions could potentially degrade natural diversity and purity, promoting engineering like principles over nature, focused ideals. The ethical aspects of synthetic biology, which encompasses potential environmental benefits, improved animal welfare, enhanced food security and human health, as well as potential reduction in human labor and disease-related suffering, are considered. There's an emphasis on biosafety, specifically the prevention of accidental introduction of synthetic life into natural environments. Ethical discussions regarding this have pointed out that biosafety doesn't solely involve biological containment, but also the protection of the public from possibly dangerous biological agent. Nevertheless, synthetic biology products don't always carry biosafety concerns or negative environmental impacts. Many synthetic technologies are benign and unable to thrive outside the lab due to their unnatural features. In considering synthetic organisms, the prevailing safety measures, risk assessment strategies, and regulations designed for conventional genetically modified organisms are deemed sufficient. Extrinsic biocontainment strategies are employed in labs and include physical containment measures like biosafety cabinets and personal protective gear in agriculture. Similar containment strategies like isolation distances and pollen barriers, originally used for GMOs, are applied, possibly offering enhanced safety control. Synthetic organisms can be designed with intrinsic biocontainment methods that limit their growth in uncontrolled environments or inhibit gene transfers to natural organisms. Such strategies span biological kill switches, engineered inability of replication or transmission of manipulated or synthetic genes to their descendants, and the use of xenobiological organisms with alternative biochemical structures. This alternative biochemistry could use artificial xenonucleic acids, XNA, instead of DNA. For instance, bacteria and yeast can be engineered to lack the capability to produce histidine, a vital amino acid. Consequently, these organisms can only grow in a laboratory setting under histidine-rich conditions, assuaging concerns about them spreading indiscriminately. Amidst the evolving field of synthetic biology comes an escalating risk of bioterrorism. As we reel from the impacts of global outbreaks like COVID-19, terrorists may potentially view this vulnerability as an opportunity to create disruption of significant proportions. This viewpoint is supported by Juan Zarate, the former Deputy National Security Advisor for combating terrorism. Securing against bioterrorism necessitates a holistic approach, encompassing crucial aspects of safety, ethics, and public dialogue. The U.S.-sponsored project SymbioSafe has identified these key issues, focusing on public awareness and continuous conversations among relevant bodies. They stress on the importance of involving synthetic DNA companies and amateur biologists. Notably, the creation of new life forms raises serious ethical concerns. However, synthetic biology presents a double-edged sword. While it can enhance the production of medical treatments, it can also facilitate the creation or modification of harmful pathogens. The biohacking community poses certain risks in this regard due to the challenges in oversight and mitigation of biosafety and biosecurity threat. A broad public understanding of synthetic biology is essential, which is addressed by SymbioSafe and another European scheme, COSI. In 2009, they released a documentary film delving into the societal implications of synthetic biology. Furthermore, the International Association Synthetic Biology advocates for self-regulation within the industry. Leading scientists from DNA synthesis companies have drafted a strategic plan for this purpose. Recognizing the broader societal implications, several institutions are engaged in the study of synthetic biology. Funded by the Alfred P. Sloan Foundation, a comprehensive examination of the public perception, ethics, and policy implications of synthetic biology were conducted by the Woodrow Wilson Center, the Hastings Center, and the G. Craig Venter Institute. In 2009, a significant symposium was convened to explore the challenges and opportunities presented by synthetic biology. Moreover, in response to the creation and swift media coverage of the first synthetic genome, a Presidential Commission for the Study of Bioethical Issues was formed by President Obama. This commission contributed to the discourse by issuing a report on the ethics of synthetic biology and emerging technologies. The President's Bioethics Commission is responding to potential security concerns arising from the use of biotechnology in the industry, specifically in the creation of self-replicating cells from synthesized genomes. They are proposing federal guidelines that focus on regulating this scientific practice and educating the public about it. Furthermore, in 2012, a significant number of environmental and civil society groups, such as Friends of the Earth, the International Center for Technology Assessment, and the ETSET Group, issued a manifesto named the Principles for the Oversight of Synthetic Biology. This advocated for a global halt on the use and commercialization of synthetic organisms until stronger regulations and safety measures are implemented. This group seeks to completely ban the use of synthetic biology within the human genome or microbiome. However, Richard Lewontin points out that despite the proposal's reasonable stipulations, the public lacks the power to enforce these recommendations. The synthetic biology field presents various hazards, including risks to the safety of workers and the larger public, possible environmental consequences, as well as threats to the biosafety sphere via potential misuse and bioterrorism. These potential risks include recreating pathogens, engineering organisms with elevated dangers, or structuring microbes to produce harmful biochemicals. On the environment front, synthetic biology poses the threat of negatively impacting biodiversity and ecosystems, and possibly inducing transformations to land use through the agricultural application of synthetic organisms. Synthetic biology's potential misuse underscores its status as a dual-use technology that could be either intentionally or unintentionally harmful. As the field is still developing, existing risk analysis systems for genetically modified organisms (GMOs) are believed to be sufficient for synthetic organisms. However, challenges may arise if an organism is built from individual genetic sequences. Despite these new territories, synthetic biology mostly falls under the existing GMO and general biotechnology regulations at the present. There are no regulations specific to synthetic biology in any jurisdiction.